All right, let's go ahead and get started. Short week. I know, it is sad. So today we do our last on molecular bio. We're going to talk about horizontal gene transfer. On Wednesday, we'll do a lecture on bacteriophages or bacterial viruses. And then we're off on class until next week, Wednesday. But we do have lab Tuesday and Thursday, and next week Tuesday and Thursday as well. Okay, so lab is unaffected by Easter break. Note that tomorrow for lab, we're doing Kirby-Bauer or disc diffusion assays. This is fair game for a quiz. Okay, so we're nearing the point where we're kind of almost all done with lab quizzes, but I just want to let you know there may be one or a couple this week, so be aware. Next week on Wednesday, we're going to have a guest lecture come in, actually. Kathleen Makielski from UW-Madison is going to come and talk about human viruses for lecture. So she has experience in HPV research, so she's going to come in and do a lecture next week on Wednesday. I think that's about it. That's all I wanted to address before we get started. So today's main goal, one objective, compare mechanisms and results of horizontal gene transfer. Okay? So let's start with a couple of basic definitions. There we go. Vertical gene transfer. What is it? When we use the term vertical gene transfer, what are we referring to? And this is a question for somebody to respond to.
Mutations?
What's that?
Mutations?
It could be mutations. Mutations could contribute to vertical gene transfer. And in general, vertical gene transfer is just transfer genetic information from one generation to the next. So mother cell undergoes replication, divides to two daughter cells by binary fission. That's vertical gene transfer. Okay? What's horizontal? How does horizontal differ from vertical gene transfer? Please somebody read off the slide. That's all I'm asking right now. I know it's Monday. I know Easter is only 6 days away.
The exchange of genetic information.
Exchange of genetic information. Without having to have replication occur. 3 forms. What are the 3 forms of horizontal gene transfer? Transformation, transduction, conjugation. Can somebody describe in a sentence or so, what is transformation? And transformation is exemplified by the one in the top left corner over here. So what is transformation? Can you speak up, Alana? Sorry. Okay, that is true for all of them. That is basically what's happening in horizontal gene transfer, is genetic information is being passed from one cell to another. The mechanism by which it occurs in each of these is a little bit different. Usually the best way to describe transformation is the genetic information is coming from the environment. So as this image up here shows, we have a cell. It undergoes lysis. The DNA genome is chopped into smaller fragments. And some cells have the ability to import this DNA into their cells. And if there is homology, or if there is sequence in that cell that has taken up the DNA that is similar or close to that of the DNA that's being brought in from the environment, we can get a recombination event. And this can introduce new versions of genes, or actually introduce completely new genes that weren't present in that organism before. What is transduction, which is the one on the top right-hand side? How is the genetic information passed in transduction? What's this little guy? What was that? Phage, or phage would be how the old timers call it, actually, but yes, bacteriophage or bacteriophage if you want to sound more formal. So bacterial virus. Bacterial virus, and we'll talk about a little bit more in detail how this occurs on Wednesday, or it may become more clear, but that bacteriophage picks up information from one cell on accident. This is kind of a mistake. Transfers that information into the new recipient cell, and again, we've got to have some sort of recombination event to occur to introduce those new genes into the host genome. Last form, conjugation. What's required to share the genetic information in conjugation? Pilus. That's the main key there. And pilus, most often, and the form that we'll talk about, is just this plasmid transfer. So an F plasmid is passed from one cell to another. It undergoes a replication during this passing so that both cells will end up with a copy of it at the time that it's done. Now, each of these serves as a mechanism for antibiotic resistance to spread. Each of these allows for mechanisms to allow for exchange of just genetic information between microorganisms. Not all microorganisms can perform each of these events. Okay? For example, E. coli is not naturally competent. And the term competent refers to the fact, refers to the question, can they be transformed or not? Bacteria that can be transformed naturally are called naturally competent bacteria. Ones that are most often naturally competent are soil bacteria. Okay. And some other human pathogens are also naturally competent as well. Neisseria, the genus of bacteria that includes Neisseria gonorrhea, which causes gonorrhea, and Zika, which is a pathogen, but to a much lesser degree, they can undergo natural transformation. All right, so after this broad overview, what I want to do for the next about 20 minutes or so, I'm going to give you a list of terminology that are associated with these different mechanisms of gene transfer. So in total, that's more than, not as many as you. In total, there are 4 different mechanisms of gene transfer. Vertical, and then 3 specific forms of horizontal. One is transformation, one is transduction, and one is conjugation. What I'd like you to do is to work with 1 or 2 other people and organize this information to the best of your ability. Some of the terms in here are specific to only one type. So for example, I think there's one that says F plasmid. That's specific to just conjugation. There are other terms that are specific to more than one type. Okay, so for example, recombination or integration into the genome, that is an example of one that is used in more than one case. All right? We'll spend about 10 minutes on this in terms of organizing, and then we'll take some time to talk about it as a group and see how we organize the information. All right? Go. Did anybody not get a copy? Okay. All right. Let's start with ones that, let's just start with what you have right now And then there are some in here that are a bit more specific or a bit more detailed terms, so we'll address those when we need to. Let's start with transformation, because that's an area that I saw a lot of people working on to start with. What are terms from this list that we might associate with transformation?
Competent cell.
Okay. Competent cell. Alana, did you have another one? DNA from environment. Good. What else? Recombination. Good. That DNA has to recombine into the genome. In other words, what this particular step means is if the genetic information coming in is not homologous, or if there are regions of it that don't look, or if it does not look at all like the host chromosome, then the recombination effect or this transformation will not be successful. Okay? So there has to be some homology. In other words, transformation works really well between two species that are similarly related to one another. For example, Staph aureus and Staph epi. We might find recombination to occur. Much less likely to find recombination occurring between a lysed E. coli cell and Staphylococcus aureus, because they're quite different from one another. Okay? What else? Grace. Nuclease is required. This is hard to read on the image, I'm well aware. But the nuclease here helps to, from this double-stranded DNA that's coming in, it degrades one of those two strands. So it produces a single-stranded DNA that, in that single-stranded form, is required for the recombination event to take place. Good. What else? There's a couple we're missing. We'll come back to it, though. Okay? This is a very good list. The ones I would definitely expect to see in here, probably the top three, are ones that I would expect everybody to know. How about transduction? Terms associated with that process. That's good. What else? Yeah, we need some sort of recombination for this one as well. Any others added to it? Those are probably the basic two I would expect for it. There's a couple missing still, but we'll come back to those. Okay. How about conjugation? Alana, get us started. Okay, so we've got the F minus, which is known as the recipient, right? That's the one that lacks the F plasmid. And then the F plus is our donor. What other terms? Pilus. F plasmid. The F stands here stands for fertility, actually. This is the closest we get to bacterial sex. All right. What else?
Replication.
Yeah, absolutely. In what way, Kelly?
I was just looking at the end, and it talks about like the F factor. Something about like, I don't know. I just remember seeing it.
Yeah, and in this image, the F factor is also the F plasmid. So this image refers to it a little bit differently. But what's happening? that F factor has to what? Undergo replication so that each of these cells, the original host and that which was F minus originally, gain a copy of that plasmid. Absolutely, replication is an important part of this process. What else is in this one that is on the list? What's that, Alan? Transfer? Yeah, absolutely. I would include that one. Requires some sort of transfer of genetic information.
The ORIT?
Yeah, ORIT. So you may remember we talked about there's an origin of replication for chromosomes. That's the point at which replication begins for a prokaryotic genome. The ORIT stands for origin of transfer. So it's this point where the transfer process occurs. And really what's happening here, if we take a look at that plasmid, it's double-stranded. In this process, what we have is one of those two strands gets nicked at what we call the ORIT, and then those two strands are separated from each other by a helicase. And once they're separated, they're both single-stranded DNA at that point. That's when the replication needs to occur to finish to reform the double-stranded DNA in both of those. Alana.
So there's a transfer, and then there's also full transfer. Can you describe it?
Yeah. So the plasmid isn't a genome per se. One of the distinctions we've made about genomes is they contain all the essential genes required for a microorganism. The F plasmid, we're kind of leaving it a bit nebulous what's on it. There's a number of different genes that could be there. One, there's always going to be a gene to encode for apillis. So the gene that encodes for apillis would be found on this F plasmid. The other things we might find here, or the ones that we're most often concerned about, are antibiotic resistance genes. So ones that, for example, we'll talk about beta-lactamases in a week or two. Those are enzymes that take penicillin and other antibiotics and cut them to inactivate them. You might find those genes on these F plasmids. The whole genome transfer refers to cases in which you have this being transferred from one cell to another. In what case, what might we see that? In which of the four cases would we see a whole genome transferred from one cell to another? Do you know, Bryce? Not transformation, actually. We'd only see part of the genome. Oftentimes in transformation, it's only part of the genome that's being transferred. Yeah, it does, doesn't it? It is vertical. Vertical gene transfer is the only case in which we actually see a full genome being passed from one cell to daughter cells. And of course, this requires replication as well. Now, to go back to Alana's question, there are times in conjugation where we will see... I don't know if I put it up on here. Yeah, I did. Where we will see part of the genome being transported as well. Those are cases in which the F plasmid has actually integrated into the host genome. We're not going to worry about this as a class, but I just wanted to make everyone familiar that there are times in which you will see a host genome being transferred during conjugation. It's in these events when that F plasmid is actually integrated into the host genome. Let's see. Let's fill out what we can for vertical gene transfer right now. So we've put in replication and full genome transfer. Binary fission, absolutely. Good. Binary fission. Mother to daughters. Or fathers to sons. Into all three in some cases. So for example, out of the terms requires homology, inheritance of new genes, genes shared across species, ATP dependent. Which of those could be terms that apply to all three forms of horizontal gene transfer? Genes shared across species, absolutely. Each of these represents an example in which we can have genetic information shared within a species, between two organisms of Staph aureus, for example, or across different species, between Staph aureus and Staph epi, or between Streptococcus pyogenes and Streptococcus pneumoniae. So that definitely is one term that fits. There's at least one other one where we might find a term applying to all three of these forms. And that's the inheritance of new genes. Each of these can lead to the inheritance of new genes to microorganisms. If we just said new forms of a gene, new alleles of a gene, that could apply to all four of these. Okay? But inheritance, a gene that was not there previously and is all of a sudden giving the bacteria a new function that has never existed before, that's unique to horizontal gene transfer. It's these mechanisms by which we have found, for example, methicillin-resistant Staph aureus. That didn't just arise out of the blue. Well, it kind of did. But it did through a horizontal gene transfer event, picking up that methicillin resistance gene from some other microorganism. Okay. Terms we didn't address, ATP dependent. You could consider all of these as being ATP dependent, probably. They all require some form of energy. The one that I most often associate with that, just to give you a heads up, is transformation. Because this bringing of the DNA into the host cell, this is an active process. This requires energy to bring that DNA into the host cell. The other one was, requires homology. Anytime a recombination event occurs. So in transformation, one of the things that we said is there has to be some sequence similarity between that piece of DNA that's being brought into the host cell and the current genome. And that similarity, the term that we use is homology. Okay? If there's sequence homology, that means there's sequence similarity between the two organisms. So we find this both in transformation and we find it in transduction as well. If there's not homology, that DNA that's coming into the host cell will actually be degraded. And those nucleotides can be reused to drive binary fission of that cell. Okay? So that's the majority of the terminology that we're associating here.
What about the ATP dependent?
ATP dependent was the one that I said I would fit most with transformation, because this process of bringing the DNA into the host cell, right here, this is a form of active transport, and that would require ATP. You could probably associate with any of the four, though, to be honest, because these all require some form of energy to drive synthesis of new DNA or to drive the recombination event. Kelly?
So are there only certain types of cells that can do like transformation and transduction, or is it like?
It's more on a species level.
Okay.
So, and for these events to take place, so for example, for transformation to take place, what does a bacterial cell have to have in order for this process to take place? Well, there's a couple of things. One, they've got to have a transport protein that allows for the DNA to come into the cell. Not all bacteria have that. So this is a restrictive step. We've also got to have this nuclease here as well. So usually we find that organisms that are able to undergo natural transformation, they have recognition of that genetic information, but also a mechanism to bringing it in and to cutting up one of those two strands as it's coming in. E. coli is an organism that will not perform transformation because it doesn't have those proteins. However, E. coli can be transduced. E. coli can also undergo conjugation. We find that transduction is a mechanism that seems to be true for all microorganisms. And the reason being is that, well, maybe one way to put it, for every one bacterial cell on the face of the earth, and there's something on the order of like 10 to the 30th bacterial cells estimated, there are 10 bacteriophage. In other words, 10 bacterial viruses for every one bacterial cell on the face of the earth. And so because of that, what we can at least assume is that each bacteria species that we know exists is going to have a bacteriophage, that's not the right image, sorry, that's going to be able to infect it. These phage have to be what we call lysogenic or temperate phage. That's a term we'll address on Wednesday, okay? But that means that they have to have the ability to integrate their genome or the host genome into itself. Okay? And since all organisms are susceptible to viral infection, we can assume that this probably occurs for all bacteria. Now, conjugation, for an organism to perform conjugation, what do they have to have? What are the characteristics of one of those donor or F-positive cells? So what does the one on the left have, the F-positive, that the one on the right doesn't have?
Origin of transfer?
Has an origin of transfer, which is a part of what, Kelly? True. What's this structure, this green structure known as?
Oh, the F factor.
Or the F plasmid,
Okay.
right? So organisms can perform this mechanism if they have this F plasmid. Not every single bacterial strain or isolate does have this plasmid. But if an F positive and an F minus cell like each other and they dock up next to each other, this is the birds and the bees talk, right? Docking, we get transferred genetic information. Now both are F positive. And so now both of these can perform that conjugation process. Each of these processes is going to more likely happen between two closely related species than they will between distinct species, but we can see it happening between distinct species as well. Quick side note, so you were asking, does all of this happen in each of these, in all bacterial species? And again, the answer is no, but there are ways that, for example, we can take E. coli and we can make them artificially competent. In other words, we can treat them with chemicals or basically send an electric pulse through their membrane that causes pores to open up and allow DNA to get in just kind of at random. So that's artificial transformation. It's not something we find in nature, but it's something that we find that we can do in the laboratory a lot. And if we're talking about like genetically modified bacteria, that's how we do it. Yeah. Any questions on these different mechanisms? Anything that's come up that we haven't addressed yet? All right. Let's take a look at a specific example of one of these. So one of the most classic, we don't talk about history too much in here, but one of the most classic experiments in microbiology was actually not an experiment to test for microbiology, but was rather a test for understanding pathogenesis, understanding does this bacteria encode that allow it to kill an individual, that allow it to cause pneumonia. And what he knew was that this bacteria was naturally competent. So what if we say that it's naturally competent, which of the pathways are we talking about? What do we know about it if we say that it's naturally competent? It can be transformed. That's what we know about it just from that terminology there. So Griffith found that there were two strains, Streptococcus pneumoniae, that appeared rough on agar plates. Those ones were non-pathogenic. So he took those rough-looking colonies, created a suspension of them, took a mouse, injected those rough colonies into a mouse. It does nothing. Mouse lives. He then took smooth colonies, injects those into mice. They die. So inject smooth into mice, they die. Rough into mice, they live. Does one more experiment. He takes heat-killed smooth, and those were unable to cause disease. In other words, the organism has to be living to cause disease. So now he takes one last condition. Takes heat-killed smooth, takes the rough, inject them together into mice, and the mouse dies. But neither of these alone, neither the heat-killed smooth bacteria or the rough bacteria alone were able to cause disease. But somehow when he combined them together and injected them, now all of a sudden they're able to cause disease. If you were able to isolate the bacteria from those mice after this infection, again, heat-killed smooth, this would mean they wouldn't grow, and he's injected rough. What would you expect to see? If you were able to isolate that bacteria, plate them out, and look at the morphology on the plate, what would we expect to see out of there? And the most likely answer is we'd expect that we'd only find rough colonies, because those were the only ones living when we injected them, or when Griffith injected them into the mice. That's not what he observed. Instead, he observed some rough, and all of a sudden this reemergence of smooth bacteria as well. How? That's what I want you to answer. So, explain, basically I'm giving away the answer, it's through transformation, but explain how that transformation process occurred and led to those results. That all of a sudden, injecting rough and killed smooth will not only kill the mouse, but we get out a mixed culture of bacteria after this process as well. Take 3 minutes. Try your best. Talk it through with somebody else. This is not an easy problem.